Dr. Miguel Sena Esteves is pacing in the back, and he's the next speaker. He's going to talk to us about gene therapy and GM1.
I'm going to change a little bit the topic from enzyme replacement therapy and chaperone to a more aggressive intervention, perhaps, or using your body to actually make the enzymes that are missing. So what we've been using for, Doug Martin, that I've worked with, I was reminded for the last 10 years that we've worked together. And so I'm going to show you work that spans a 10-year period, which, once you realize it, you also realize you're getting a little old. But anyway, so Adeno-associated viruses, actually, one of the simplest virus that you can have. It's really small. It's 20 nanometers. I mean, you can think of this as a nanoparticle, actually. And in fact, it's so simple that it only has 2 genes. So this is as simple as it gets in terms of in the viruses category. This is one of the simplest ones. And part of the reasons that we like to use it is that if we took blood from everybody in this room, about 80% of us have been exposed to it, and there's no disease associated with it. Okay, so as a starting point, that's great because you don't have any disease associated that you have to worry about. So it's certainly a great platform for that. But more importantly than that is that I can change the slide. Okay, all right, well, there we go. Is that you can actually, people, about 30 years ago decided that this could be an interesting platform to develop gene therapy. And thanks to the foresight of those investigators, there were actually 2 in the entire United States and in the world, actually, that decided that this was a great idea. Turns out 20 or 30 years later that they were dead on the mark, and in fact this has become sort of the most promising platform for developing gene therapy approaches. So the trick in the next 20 years of work was to figure out how to take out the viral genes and put inside the information that is necessary for a cell to make a protein, so to make a gene, to bring a normal copy of a gene into the cells. So part of the reason that we really like these entities that we just simply call AAV is that they are extraordinarily efficient for gene delivery to the vast majority of organs throughout the body. So if you put this IV, you can turn the liver, deliver gene to the entire liver or to the muscle, and I'll show you what we're able to accomplish when we inject them into the brain. And in the second part of the talk, I'll show you what we can accomplish if we put this through the vasculature. So there are several aspects of it that are quite important. One of them is that it doesn't integrate into the cell genome, which means it's very unlikely to cause secondary effects of the gene transfer itself. And importantly, once you've delivered it, the data that we have right now is that in principle is going to be expressing the gene for the duration of the lifespan of the organism. So we have data from monkeys, not as in particular, but the field has data in monkeys that 15 years later they're expressing just as much protein as they were 1 month after injection. So that's pretty cool because it opens the possibility that one treatment is all that you need, okay? But then again, we have to learn from humans whether that is the case or not. So in terms of the brain, or access to the central nervous system on how you can deliver drugs, there's a number of approaches that you can consider. One of them is that you take a needle and stick it straight into the brain. That's one way to do things. The other one is that you can go into the cerebrospinal fluid, which is mimicked here, and you inject viruses or proteins, and that's being explored actually for enzyme replacement therapy for neurological or for lysosomal storage disease with neurological features. Or more recently, we found out that, in fact, as Carol was pointing out, that some viruses or some AAVs can actually cross the blood-brain barrier, so opening the possibility that in addition to these approaches, direct injection into the brain, we may be able to also develop therapies based on simply an infusion into the bloodstream. So work that we started about 10 years ago in mice showed us that if we injected an AAV vector into a very specific structure in the brain called the thalamus, which is located here, what was a bit of a surprise for us is that you could express the gene in this location where you injected it, But in fact, what we found out, and this is a month later, so it happens really, really, really quickly, is that the enzyme spread throughout the entire brain. And this is the front of the brain all the way to the back. So blue means there is a ton of enzyme in it. And if there's no blue, well, it means that there's no enzyme. And this was done in a GM1 gangliosidosis mouse brain. So this opened the possibility that instead of having to stick 1,000 needles into the brain, if you choose the structures carefully, you may actually be able to distribute enzymes throughout the entire brain without having to go and turn someone's brain into a porcupine, which of course carries quite a bit of risk. So this is a strategy that we recognize this as a way to translate from a small mouse brain, which I work with mice all the time, so it's really small. I mean, compared to the human brain, it's 2,000-fold smaller. So you can imagine that whatever you do in the mouse, just think of something that has to be 2,000-fold, either more injections or bigger, whatever way you want to think about. So the notion of having a few sites treated and being able to actually affect gene delivery or protein delivery to the entire brain was quite attractive to us. So we initiated some studies in GM1 gangliosidosis mice, and you can see that we express boatloads of this protein out of an AAV. This is looking at the brain 1 month after infusion. And what you can appreciate here, this is looking at enzymatic activity. This one is the activity in a normal mouse. There's barely anything detected in these black bars that are the untreated animals. And within a month, we can have very high levels of expression of enzyme throughout the entire brain and spinal cord. And this happens very rapidly. And importantly, what we also found out is that it doesn't change. You can see that if you looked at 1 month, you see these levels here. 4 months of age continues at about the same levels, or a little higher, and 6 months later, it's still at the same level. So giving the notion that whatever you achieve is permanent, which of course is quite important for an intervention that is pretty invasive, because this is an injection directly into the brain. So clearly, you don't want to do it once and a couple of months later having to come back again and again and again. So the notion that it's for a long time is quite important. Also, obviously, getting enzyme into the brain is quite important, but more important than that is the biochemical changes. So GM1 ganglioside, which is up here, so this is just a way to measure the content in the brain of this ganglioside. And this is a level that you have in a normal animal, and you can see here that it's elevated quite dramatically in untreated animals, and within a month, we've restored it back to normal. Okay, so it's quite exciting that you can have, within a month, a very dramatic response to this type of therapy. And so based on some of these results, this is just a survival. I'm going to skip that, but essentially animals died here, and we stopped the study at 1 year of age, and the animals were doing pretty well. So based on these studies that we've done initially in mice, so the notion of what we want to translate into the humans is that we're going to do direct injections to very specific locations, in our case the thalamus, to actually get the enzyme to distribute throughout the entire brain. And so 10 years ago, someone with foresight put Doug's poster in a meeting right next to mine. So whoever that was, actually very smart, clearly. And we started talking because, he works in cats, I work in mice, and, well, it's pretty obvious that going from mice to cats is a nice intermediate step before you get into a human. So we decided to implement the same strategy. In the way, there's lots of stories about things that, well, didn't quite work out as we expected. Mice, immunologically, the immune system of mouse is kind of funny. You can do a lot of things to them. Once you move to other species, the immune system is a lot less tolerant than a mouse immune system. So we learned that the hard way, but then we started to do experiments where we put the feline enzyme, or the cat enzyme, into cats, which, ultimately you want to put in a human is a human, not from another species. And what we found was exactly the same, which is pretty cool because, again, it tells you that the biology that you found in a mouse is not just, funny mice, funny biology maybe, but it turns out that within a month, again, injection into a single site in the thalamus distributed enzyme throughout the entire brain once again. And yesterday I showed data about exosaminidase, and it's the exact same thing from mice to cats to sheep. Obviously, there's no guarantee that in a human that would be the case, but certainly having it across three species where the brain size keeps on increasing is quite encouraging. So this is what you would see in a normal animal, and there is actually here the amount of activity that is in the GM1 cat, which is nothing. So just to cut a long story short that lasts, well, about 10 years, I'll just show you some videos. This comes from Doug's lab that has done all this work in cats. And essentially, these cats got 4 injections. That's it. We went from the mouse with 4 injections to the cat with 4 injections, and our plan is to actually move to the human. As a matter of fact, to the human actually with 3 injections. So you really don't plan on injecting 1,000 sites to be able to achieve a therapeutic effect. And the results actually were pretty stunning, because we went from trying many things for many years, not being able to change at all the outcome in animals, to all of a sudden Doug starts calling me and tells me, well, these cats are doing fantastically well. And we eventually got an NIH grant. And then the problem was that these cats were all alive. And the grant ran out. So poor Doug there, I don't know how he raised the money, but he kept them alive for a long time. So of course that's always a good problem to have. But certainly you can appreciate here that these AAV, and this is not the only one, the success was not exactly the same in every single one of them, but a very large number of them survived for a very long time. So based on these results, we're actually quite excited that in 2015 we started working with Lysogene. So it's a company in Paris. Sam, that is here, is a representative of Lysogene. And so we formed a partnership to move this towards the clinic, because that's certainly the next step. And working with a company that has gone through a phase 1 clinical trial means that there is expertise to help us get through the next steps, where it becomes a little bit outside of our expertise, because we're an academic. I play with mice, Doug does cats, and it's a little bit different than when you get to the humans, right? So having a partnership with a company that has done this before is a huge advantage for everyone, and I believe for the patients as well. So Lysogene is going to lead the clinical development piece of it, which we're ecstatic about that for sure. The vectors that we're using is a little switch from what we've done before. We're using AAVrh10. There's many flavors of AAVs. This is one of them. And the reason we chose this one is twofold. One is because the first trial that Lysogene ran used AAVrh10. And moreover, this has been put into patients, into the brain, in a number of diseases, which of course the regulatory hurdle, meaning for the FDA to accept this as a clinical trial, is a little easier than if you come with something that they've never seen before. So we made the decision to move to this AAVrh10, and what we're doing is optimizing the design of these vectors, both working still in my lab at the University of Massachusetts Medical School and at Auburn University. So in mice and cats, again, we continue that ping-ponging between the two species that has been incredibly informative. The plan is to have a pre-IND with the FDA to tell them, look, this is what we want to do. What do you think about it? Essentially, that's what a pre-IND boils down to. This is our plan. Then they tell you back, well, we either agree completely, but usually, they come back with comments about other things that they'd like to see. And the plan right now is to run a phase 1/2 sometime in 2017 to 2018. I certainly hope more for the 2017 than the 18, but you never know. But certainly between 2017 and 18, that's the plan, to have a phase 1/2 clinical trial, which I think the plan right now is to do it in the United States. So now I'm going to change a little bit into a second part of work that Doug and I have started to do, which is an alternative approach that I already mentioned to you, which is going through the systemic administration. So essentially, put it through the bloodstream, and let's hope that it gets into the brain. So this is actually a picture. So someone decided that this is a pretty cool thing. I love this picture. I'm a geek, I admit that. This is a plastic cast of a brain vasculature. So this is how many vessels your brain has, okay? Which is pretty incredible. So if you think about it, here in this little space between all these little tubes that are the vessels, it hits about 2 cells. So every cell in your brain is incredibly close to a blood vessel, okay? So if you think about it, I mean, this is the perfect way to deliver drugs to the brain, right? But as Carol pointed out just before me, there is a little detail that the brain likes to live in its own little environment. And the way that it does that is through a blood-brain barrier, which is a pretty complex structure. Wow. Certainly changing slides is one of it as well. Okay, back one. Thank you. So as Carol pointed out, you know, the blood vessels, yes, they look a little bit like the rest of the blood vessels in your body, but the brain has developed, the vessels in the brain have developed a complex structure that regulates very, very carefully what gets from the circulation into the brain. I mean, after all, think about this, right? Your brain works on electrical activity. Imagine that at least for that reason, it needs to live in an environment that might be slightly different than the rest of your body. So essentially what this does is that it prevents the vast majority of molecules that you take, or that viruses or drugs that you take from getting from this inside of the vessel into the brain itself, okay? So as I showed in the slide before, one of the approaches that we've developed over the last 10 years, and we're going to move into a clinical trial, is a direct infusion, and now we're looking at also how effective might it be to use some of these new AAV vectors that can cross from the blood into the brain. And so a seminal paper in 2009 demonstrated for the first time that AAV9, again, you hear these numbers, right, and it doesn't mean much, but so when you hear AAV9, what Kevin Faust and Brian Casper showed for the first time is that if you put these vectors into a mouse, into a neonatal mouse, or even an adult, what you find is that green here is just a gene, right? So we don't fluoresce, our cells don't have this kind of stuff. So this actually comes from a jellyfish, that the protein is green, and it's very useful for us. Actually, a Nobel Prize was awarded because of the discovery of this protein, because it has revolutionized biology as well. So we use this a lot in the lab to know where are we expressing genes. So essentially what they showed is that if they infuse this into the bloodstream, they could see genes being expressed in the brain. So this actually has revolutionized the gene therapy field quite dramatically. And what we found over the years since then, since 2009, is that this happens in monkeys as well. So here is a normal monkey. The gene that is being used to look at the expression is not there. And when you infuse the virus through the circulation, this is what you find. So it's expressed quite well in different places. And in fact, our colleague Wenping Gao at UMass Medical School actually started to look at capsids other than the AAV9, and it turns out that its property of getting across the blood-brain barrier is not unique to AAV9, but in fact there is a number of other AAVs that can do the exact same thing. So in this landscape, we decided to ask the question as to how effective might this be for GM1 gangliosidosis. So we injected an AAV9 vector encoding the beta-galactosidase that you've already heard about today that is missing in GM1, so a normal copy of the gene, and infused these mice at about 6 to 8 weeks and started to look at enzyme expression throughout the body, including, of course, the brain, the cerebellum, spinal cord. And what we find, again, very much like what I've shown you before, is that you have here very low activity levels in the untreated animals. A normal level is here. And what we're able to do, at least in the central nervous system, is increase expression to about 50% of normal, simply by a vascular infusion. And in other organs where these viruses get into with much, much higher efficiency, then the elevation in enzyme activity is in the order of 100 to 200-fold over normal activity. What we found is that although we had a clear increase in activity, the effect was not as potent as we had with intracranial injection. And so up here, so this is an untreated animal. If you see little dots, little white dots, means that there is storage, means that there is the GM1 ganglioside in the brain. And what you really want to see is essentially this, which is a black background, which means that you've cleared storage. Okay? So what you can see up here, it's in one of the treated animals we've reduced it very dramatically from an untreated animal, but is not completely cleared, okay? And this is the quantitative data that we've done, and in fact reflects there very nicely. This is an untreated animal, this is the normal amounts, and what we have here is a reduction in the content of GM1 ganglioside in the central nervous system. One of the hallmarks of GM1 gangliosidosis, and in fact Sandra Dazo has published very nice papers on this, showing that in fact an inflammatory response is part of the disease progression. So this is a marker. So some of your cells, as the disease progresses, they're not happy. There is toxicity, and so they start to respond, they start to react, okay? And we can look at the brains of these animals and use some specific stains to see how are those cells present or not. And so this is in a normal animal, so you don't see them that much. You can see that these cells, so this stain here is very, very intense in a treated animal. And what you can appreciate is that in animals that were treated, and this is a male and a female, there is a marked reduction in these cells that are not too happy in an environment where there is some degree of toxicity. So clearly, we have changed quite a bit the outcome in these animals with the vascular delivery. And this is sort of brain weights, body weights, we're not going to go there, but just in terms of survival, what we found is that we could extend survival from about 8 to 9 months of age out to some animals past a year and a half. And in fact, in females, it worked quite a bit better than in males. We could discuss that a little bit later, but what we found is that these systemic deliveries work better in females to get into the brain than in males. We really have no idea why, but that's an observation that us and other colleagues have made. We have no idea if this would be true in humans as well. So what I've shown you here is that, well, in fact, you can actually achieve some degree of treatment or some degree of correction in the central nervous system by an infusion into the bloodstream, but it's not perfect yet. To get to a full correction, we probably would need doses that are very, very high, and when you translate from mouse body weight into a human body weight, then we're start to get into an issue of production. And you've heard before some people talking about production of a chemical, production, and then we're talking about production of these entities, which is rather complicated and extraordinarily expensive. So the scale and how much you need to achieve a therapeutic effect actually matters quite a lot. So in my lab, over many years, what we've been trying to do is to improve on the capsids that are existing. And one of the approaches that we've investigated is take one of these AAVs, and this is sort of representation of what it might look like if you looked at it really, really, really close. And we started to decorate its surface with different types of peptides to try to make it cross the blood-brain barrier better. And what I'm going to show you here is the difference between an AAV9, which I've shown you the results before, and one of the new capsids that we have. An animal that was not treated with one of these would be completely blank. So what I'm showing you here, the staining, means that there were genes that it was transferred into tissue, it's expressed, and we're using a stain to detect it. And you can appreciate the difference between an AAV9 and one of these new capsids, and it's quite exciting for us as well, is that not only we have this massive increase, but in fact, while in a normal, with an AAV9, we find a lot, these little squiggles here are either astrocytes or they're blood vessels. What we find with the new AAV is that these cells here that you're finding or seeing here, so this is very important not only for GM1 gangliosidosis, Tay-Sachs, but in fact, this is critical for the types of neurodegenerative diseases that we're also interested in and we work on as well. So for us, this is a very interesting step forward because in two ways, is that may give us access for the types of neurodegenerative diseases for which these cross-correction mechanisms of lysosomal storage disease don't necessarily work, but also importantly for GM1 and Tay-Sachs, allow us to achieve a complete correction of the phenotype using much, much lower doses than we would have to use with AAV9. I think this is only a fraction of the people that have worked over this, on these projects over the years. You can appreciate here that to do experiments in large animals it's almost a small village, really. And so none of this, of course, is possible without funding. We've been very lucky with the funding that we've gotten from the NIH, 2 different institutes at NIH, for which we are very thankful, and all of this work so far has been possible due to this. And now we're getting quite a lot of help from the Cure GM1 Foundation to try to translate some of these new molecules and new AAV vectors from these initial marking studies and start testing them in GM1 mice. And as you've heard yesterday, Doug and I received another NIH grant to study some of these new entities in Tay-Sachs disease as well. So with that, I'll stop and I'll be happy to answer questions.
I have a question from someone who's streaming online. So they wanted to know if you have any thoughts yet about what who the criteria will be for who might be a candidate for a clinical trial. If you thought about where in the disease course, newly diagnosed versus later progression.
Right, so I think Cindy might be the best person to answer that question, because we're hoping, well, we're all punting the answer, so all right, I'll give you part of the answer and then Cindy can give you the rest of it. So based on the studies that we've done in animal models, right now the most straight up answer we can give you is that the best we can hope for is to stabilize disease progression, okay? One of the goals of a clinical trial is try to find 2 things, right? One is a safety concern, right? So is the thing that we're using safe? But in gene therapy, especially for a deadly disease, no one carries out a trial that is a pure safety trial. You're carrying out a trial to know, yes, is it safe? But you're trying to learn whether there is efficacy, and based on some of these animal experiments, the expectation is that yes, we will see some efficacy. So the thing is this, is that if you choose patients where the disease has progressed quite dramatically, you run the risk of not learning anything at all, okay? So you may say, well, I didn't kill the patient, but I didn't make any difference. So in the end, essentially, we gave someone an expectation of success that will never be met, at least based on the animal experiments. If an animal is too far gone, our experience so far is that it has absolutely no effect, okay? However, I would also caution there that human development and animal development occurs at very, very different paces. So we could be in for some surprise in the human, but I would like to set people's expectations that if the disease has progressed, to a point where the patients can't even sit by themselves, the likelihood of us being able to reverse it, I would say right now is about 0, okay? So if we actually want to learn something out of these clinical trials, you have to choose patients that have some degree or a great degree of motor function and cognitive function that you can then track very accurately the decline, or hopefully no decline whatsoever, and actually improvement. And Cindy just showed you a number of measures by which to assess that, okay? And that's why this is absolutely critical, these type of studies that Cindy has done for GM1, which is very fortunate that she's done them. So while we haven't discussed exact criteria, inclusion-exclusion criteria, you've understood, I think now by what I've told you, that patients will have to have some very high degree of functionality to be enrolled in these first clinical trials. In terms of, what are the forms? Look, the reality of it is that in the infantile forms, the moment they're diagnosed, usually is because they are not meeting milestones any longer, right? So in a sense, you can think about it that infantile forms probably are not going to be in the first patients that are going to be selected for this. So most likely, and this is discussions that we've had, is that patients that are being enrolled in that late infantile, juvenile forms where the disease progresses a little slower, they're diagnosed a little earlier, and of course when they're diagnosed, yes, there is obviously some effects of the disease, but clearly they're still functional. So those, in my opinion, are probably the patients that are going to be enrolled in the first trials. It's not to say that if things work, that we're not moving very rapidly to other forms of disease, of course, because certainly we want to be able to help as many patients as we can. But first and foremost, we want to prove that it's safe and that it can make a difference to patients. Is that Cindy?
Yeah, I mean, and we've had these discussions with the FDA over other issues. If you were the FDA, you want to do everything first in adults. And they came back at us at the beginning and said, why can't you do this in adult patients? Move away. Why can't you do this in adult patients? And we said, well, it's a risk-benefit thing because we actually know that adults progress very, very slowly. Well, it's actually two things. It's adults progress very, very slowly, and they change slowly over time. So, it might take you 10 years to actually get an answer. And if you're talking, as Miguel was at the time, we don't know how this is going to work now. Is this going to be an intravenous treatment? Is this going to be an intracranial injection, a direct injection into the brain? That's fairly risky. And that where their disease is not survivable, families are willing to take those kinds of risks. If there's nothing else available, you say, well, what is my choice? So that families are really actually willing to accept more risks, to be honest with you, than the FDA is willing to allow them to accept in many cases. But in the adult situation, we thought, well, what if you did this introduction and you actually induced a stroke or something like that? Now, the adult patients would be fairly functional for decades, let's say. So we pretty much came back at them and said, well, this is really not an appropriate population to do the studies in. And so we were trying to bring it to a point where it was patients who were fairly high functioning, but in whom, with outcome measures that were carefully monitored, we could get an answer in the shortest possible time. Because, number one, you have to get, if you're going to do the trial, you want it to succeed. So you're going to optimize your ability to have it succeed as rapidly as you can, because the faster you can get the first trial done with a good outcome and a good result, the faster the FDA is going to approve it, and once it's approved, then it could be used for everyone. I mean, that's kind of the bottom line.